Hi everyone, my name is Jefferson Silva. I'm from Federal University of Pará in Brazil. Me and my co-workers hope you like our audio slide presentation. In these slides, we will talk about a helmet, a nematode specifically, called Ortleppascaris, which parasites an amphibian, the Rhinella marina, in the Amazonian region. The biological life cycle of this nematode remains not elucidated, but we know that the adult parasites can be found in crocodilians, the definitive hosts. However, the larval stages are found in some amphibians, like Rhinella marina, the so-called kinetode. These parasites also develop a fibrocysts, the white structures that can be seen along the liver at the right side on the screen. In these two pictures, we show the difference between infected and non-infected livers. The infected one presents some huge alterations. For example, in the file center, a fibrocyst containing one larvae, inflammatory infiltrates pointed with a black arrow, decrease of the blood vessels, and increased number of important immune cells, the melanomacrophages, that can be seen as the black-brown cells along the tissue. Observing this infection, a question remains: how these huge changes can occur without the death of both organisms? We believe that some molecules present in the larvae can play an important role in the maintenance of this infection. These molecules can be found in parasite surface, digestive enzymes, secretory-excretory products, or even from intestinal contents. So, the goal of this article was search proteins present in the body of the Ortleppascaris that can be involved in the host-to-parasite relationship. To achieve these objectives, we have used two-dimensional electrophoresis and mass spectrometry. In our results, we could characterize 60 spots related to different proteins seen in this electrophoresis gel. The molecules detected were classified into different biological processes. For example, the green marked spots shows proteins involved in metabolic process for resisting to the oxidative stress and evasion of the host immune system. Inside the red circles, we show the structural molecules that can be participating in organic morphogenesis. Among them are cuticular collagen. This molecule can be involved in antigenic variation on the nematode surface. Into the black ones are presented immunoreactive proteins and virulence factors. In yellow were pointed molecules of biosynthetic and secretory pathways. Into the blue one are molecules involved in the sensory process. In conclusion, some proteins identified in Ortleppascaris are highly evolutionary conserved. In this present study, other important molecules of helminth biology were found, such as immunoreactive antigens specific to nematodes, and virulence factors that are considered potential vaccine candidates, and protein markers for helminth infection. As we know, the helminths are complex and widespread pathogens, and today can affect about 2 billion people around the world, being an important public health problem. And because of it, molecular studies are needed for strategies for future interventions. The proteomic approach used here provides clues about the host-parasite relationship at the molecular level. Thank you for your attention and enjoy the article.